Nucleotides. Nucleotides are the building block of the DNA. A nucleotide consists of three components: nitrogenous base, pentose sugar, and phosphate group. There are two types of the nucleotides. One is called as oligonucleotide and the second one is called as polynucleotide. The meaning of the polynucleotide is that a long chain of the nucleotides which are joined with each other. On the other hand, nucleoside is the combination of sugar and base. If we see the nucleotide monomer, we can see that there are three components. One is the pentose sugar. At carbon number 1 of pentose sugar, nitrogenous base is attached. This is carbon number 1. And at the carbon number 5 of the pentose sugar, phosphate group is attached. So these three components collectively, nitrogenous base, phosphate group and pentose sugar collectively is called as nucleotide. Within the nucleotide, there are two types of the sugar molecule. The sugar molecule which is present in RNA is called as ribose sugar, while the sugar molecule which is present in DNA is called as deoxyribose sugar. In nucleotide, the nitrogenous bases, there are 5 different types of the nitrogenous bases: cytosine, thymine, and uracil. These are single ring nitrogenous bases and collectively they are called as pyrimidines. On the other hand, there are 2 additional types of the nitrogenous bases. Those nitrogenous bases are called as purines. Purines are adenine as well as guanine. Adenine and guanine both are 2 ring. We can see in the diagrams that adenine is a 2 ring structure, while in case of guanine is also a 2 ring structure. What is the meaning of polynucleotide linkage? If we see this diagram, this is one nucleotide. This is the second nucleotide. So nucleotide 1 is attached with the nucleotide second and second is attached with the nucleotide number 3. So polynucleotide means the nucleotides which are the building blocks of the DNA, they are joined with each other. One is joined with second, the second is joined with the third nucleotide. There is a linkage between nucleotide 1 and nucleotide 2. The linkage between nucleotide 1 and nucleotide 2 is called as phosphodiester bond. So we can say that the nucleotides within the structure of the DNA, they are joined by a bonding. The name of that bond is phosphodiester bond. In this diagram, we can see that how there is the pairing between adenine and thymine as well as between cytosine and guanine. This is the structure of the thymine and this is the structure of the adenine. There is a double bond, bond number 1 between these two bases, thymine and adenine, and this is the second bond between thymine and adenine. While on the other hand, there are 3 bonds between cytosine and guanine. In case of cytosine and guanine, there are 3 bonds: bond number 1, bond number 2, bond number 3. So In case of adenine and thymine, there are 2 hydrogen bonding, while in case of cytosine and guanine, there are 3 hydrogen bonding. We can conclude that nucleotides are the building block of the DNA, and nucleotides are composed of 3 components.